Students, so after our prac with the strawberries, I'm just going to give you a rationale or just an explanation of what happened and why it happened and why we use strawberries and detergent and salt in our solutions and of course alcohol at the end. So in our experiment we used salt, detergent and alcohol. And the alcohol that we used was ethanol. Probably wondering why we use these. Well, first of all, strawberries. Strawberries are what you call octoploid. They in their chromosomes they are octoploid. Octoploid, oct means 8, and ploidy in genetics means sets of chromosomes. So they have 8 sets of chromosomes. 1, 2, 3, 4, 5, 6, 7, 8. Humans are actually diploid. Diploid means di means 2. So humans have 2 sets of chromosomes. We've already been through this in the other lessons prior to this one. So that's the reason why we used strawberries. They have a lot of chromosomes, a lot of DNA, and that was evident in our results. Now let's get to the first reason. So after we mixed our solution, we used a detergent. Detergents look like this. They have a polar head and a non-polar tail. What that means is the head is hydrophilic, sorry, and refer to the word hydro meaning water. Philic means loving. And the tail is actually hydrophobic. Hydro meaning water. Phobic means scared of or hating. So phobic means hating, philic means loving. So the heads of the detergent molecules are polar and they are hydrophilic. The tails are hydrophobic. Why is this important? Well, when we have our cells, the membranes look like this. And they are a very similar structure. The heads are philic and the tails are phobic. When they meet, once they meet, the phospholipid bilayer, we'll get more into what that term means a little bit later, probably in the next few years of science. The heads break, and hence exposing the organelles in the solution. So once this has happened, all the organelles have been like burst. So if this is a cell, the cell has burst out like this. You have the nucleus, you've got the ribosomes, the fluids in the solution, you've got the reticulum all just going out into our solution. Okay. Now that the DNA is into our solution, we have our salt. And what salt does, NaCl, slightly negative. DNA is negatively charged. Okay. Salt allows the DNA to gather together into the sample and eventually clump together because of the charges. That's what salt does. So salt just allows them to just come together and group together, and then we need to separate them. And what did we use to separate them? We used the rubbing alcohol. Okay, the alcohol then. Now we had the two layers. We had the solution, and then we had the alcohol. And then our DNA. And that's just a basic explanation of what happened in today's prac.